Hello everyone, my name is Anna Salvian. I'm a PhD student at the University of Surrey in the United Kingdom. Today I'm going to show you some of the work that I presented at the North American ISMET conference that took place in Houston, Texas at the beginning of April 2024. I really wanted to express my gratitude to EBNet for providing the funding to being able to attend the conference. And now I'm happy to share my work with you. I hope you will enjoy it. My research is focused on the development of microbial fuel cells-based biosensors for biochemical oxygen demand monitoring. And in particular, I'm trying to move from lab scale to real applications. In the context of wastewater treatment plants, one of the main issues is the energy required to perform the treatment. Just to give you an idea, the amount of electricity used by wastewater treatment plants in Europe is higher than 1% of the total electricity used, and this percentage goes up to 3–5% in the US, so that's a huge amount. The step that requires the highest amount of electricity is called aerobic treatment. Here, oxygen has to be supplied in order to clean and treat the wastewater, and here is where the BOD biosensor comes useful. So the amount of oxygen that has to be supplied is proportional to the concentration of the BOD of the wastewater that has to be treated. Now, the main problem is that nowadays the only method to measure BOD takes 5 days and specialized personnel. So this hinders the possibility to optimize and automatize the wastewater treatment process. So in the past 15 years, researchers try to exploit electroactive bacteria in order to measure in a fast, reliable, and self-powered way the BOD of the wastewater, and this could lead to an enhancement of the wastewater treatment process, so an optimization. An MFC biosensor works as a classic single-chamber air cathode microbial fuel cell, where bacteria are on the surface of the anode and they act as a catalyst for the oxidation of organic pollutants, while at the cathode there's the reduction of oxygen from the air, oxygen to water. This oxidation at the anode and reduction at the cathode creates an electrochemical potential difference that enables the electrons to flow from the anode to the cathode when we close the circuit with an external resistance. Now, the higher is the amount of organic pollutants present in the wastewater, the higher is the current that we can measure across the external resistance. And this is the principle behind the biosensor. During my research, I tried to answer mainly two questions. The first one is, can a lab develop MFC biosensor, so with a biofilm that was created in the lab, be directly applied in wastewater treatment facilities? And the second one is, we know that bacteria, especially when it's a mixed culture, adapt to different environmental conditions, so different temperature, different substrate. So how fast does the Nordic biofilm adapt to urban wastewater, and why? So in order to answer to those two questions, I created an experimental plan that started with the inoculation of the biosensor using the sediment of a lake that we have in the university campus at the University of Surrey. So it's an environmental inoculum. The whole experiment was carried out in batch mode, and the first 2 months the feeding was just pure acetate, and then more complex synthetic wastewater, and finally urban wastewater that was not sterile. I didn't adjust the conductivity, I didn't purge with nitrogen, so it was urban wastewater without any pre-treatment. And I tested the biosensing capabilities of the microbial fuel cells with the three substrates, and I particularly focused on the complex synthetic wastewater and urban wastewater during the whole experiment. So long story short, both with complex synthetic wastewater and urban wastewater, we got a good linear response and a good accuracy. of the biosensor, showing that it can be used with different types of substrate and it can be moved directly from a synthetic lab-controlled environment to real wastewater treatment environment. We did some taxonomic analysis of the anodic biofilm to understand whether there were any fluctuations of the relative abundance of the taxa and if new taxa were incorporated when feeding with real wastewater. The answer is that there are fluctuations of the relative abundance, but the main three species remain the most abundant ones, also like when feeding over time with real wastewater. And also we found out that the bacteria present in the urban wastewater couldn't manage to incorporate in the anodic biofilm. So at that point we asked, why did this happen? And to answer this question we did some metagenomics analysis. And in particular, we measure the genes present when on the biofilm when feeding with synthetic wastewater and when feeding with urban wastewater. And we found out that when in urban wastewater biofilm, there are less genes for simple molecules degradation, such as sugars, and more genes for degradation of complex molecules such as xenobiotics, toluene, butanoate, propanoate, which are more present in the urban wastewater and weren't present in the synthetic one. And also we found out that there are more genes for antimicrobial production resistance, which could explain why new taxa were not found in the biofilm after feeding with non-sterile urban wastewater. And in particular, the biofilm was found to be able to produce non-specific antibiotics such as vancomycin, penicillin, and cephalosporins. So concluding, MFC's biosensors can be used without any pre-treatment of the feedstock, and there's no need to wait for biofilm adaptation time because the biofilm is very robust. And if there are fluctuations in the taxa, this is because the biofilm adapts to a new chemical and microbiological composition of the feedstock. Future work involves the test of MFC-based biosensors in continuous flow and during different times of the year to address any seasonal variation of the wastewater. I wanted to thank my supervisors and colleagues from the University of Surrey and from the University of Alcalá. And a big thank you to ISMET for giving me the possibility to present to such an important conference. Thank you. Bye.